Good morning, everybody. So for those of you who didn't meet me yesterday, I'm Day2T1 Behbehani. I'm one of the senior hem-onc fellows in the lab. And most of my work is focused on myeloid malignancies, leukemia, and MDS. And what I'm interested in long-term is how do these things respond to the therapies that we try and treat them with. But one of the things I developed to get at that question was a method for doing cell cycle analysis by the CyTOF, and that's what I'm going to talk to you today about today. And this is mostly just sort of a methods talk. I published all of this in Cytometry in 2012. So if you want to look for that paper, I believe it was the July 2012 issue. It basically has all the information that I'm going to talk about today, so don't worry if you don't keep up with your notes. So I'm going to talk to you a bit about a method for cell cycle analysis, markers that we use for S phase, G0, G1, G2, and M, and the validation experiments I've done with cycling T cells. Then I'll go on to talk to you about sort of a system-level analysis for doing cell cycle in normal and malignant hematopoiesis, which is sort of the main application I've been focused on. So you may know already that you can do DNA staining by the CyTOF, and that if you've got a flow background, you would say, well, why can't we just do cell cycle that way? And this is the iridium intercalator that we use for that. It sort of has this group that intercalates between the strands, and it chelates an iridium atom there. And it mostly is used in the CyTOF as more of like a forward scatter to give you a sense of the size of the cell and that there's a cell there. And you can see the curves that are generated. So this is like a Hoche stain of DNA detected on fluorescence, and you can see this sort of G1 peak, sort of S phase region in between, a distinct G2 peak. And with this, you can get a rough sense of cell cycle by gating on those peaks. But if you look at the iridium intercalator, you can see that there's really just sort of one peak. Interestingly, once you know the cell cycle state, and I'll tell you later how we get to this, but you can see that it does increase as the cells progress through cycle, but it's just not anywhere close to something that you could gate on. So we needed a different way to approach this. So what I focused on was iododeoxyuridine. So you probably are familiar with bromodeoxyuridine. This is essentially the exact same molecule, except instead of an iodine here, we have a bromine. So it can incorporate in DNA in the place of thymidine. And then the beautiful thing about IDU and the CyTOF is that instead of in BRDU, where you need to do something to open up the DNA, like acid treatment or DNase, and then come in with an antibody, we don't have to do any of that. We can just detect the iodine directly. So once it gets incorporated into the cell is labeled. When it gets vaporized in the plasma, you can detect the iodine that got incorporated in essentially immediately. And so even with as little as a 5-minute labeling, you can pick out a distinct S-phase population. And as you go to longer labelings, you see there's more IDU per cell, but the fraction of S-phase cells doesn't really change very much. You know, the slight increase when we get out to 60 minutes probably reflects just more cells that have come into S-phase during that time. And so we compared this to sort of fluorescence cytometry. So if you look at the same cells by fluorescence and by mass cytometry, again, you can see essentially similar populations. This works at concentrations of iododeoxyuridine that are pretty standard. So the sort of 10 micromolar is pretty much the standard, and you can see that in both cases in fluorescence or by mass cytometry, you get a pretty clear positive population with a 15-minute staining with 10 micromolar. So that left us needing a way to define the other phases of the cell cycle. And the key thing was that we now had a really good marker for S phase. And so once we knew S phase, it made it a lot easier to find other phases of the cell cycle. So we wanted to focus on markers of the G0 to G1 transition, markers of G2 versus G1, and markers of M phase. And we essentially were able to find markers of all of these things. So we screened a bunch of different molecules, and the ones that turned out to be most useful were cyclin B1. So again, if you look at just cyclin B1 in general, you can see that as the cells progress through cycle, it increases. But once you can pull the S-phase cells out, you can see a distinct separation. So here are cyclin B high S-phase negative cells, cyclin B low S-phase negative cells, and then the S-phase cells in between. So once you can pull these cells out of the middle, the S-phase cells, then you can define these distinct populations. If we look at phosphorylated RB, this is a site 807, 811 that gets phosphorylated during the transition from G0 to G1, as opposed to what you're typically used to as a G1 to S phase transitions. And there's a Cell paper about this from like 2008, I want to say, or 2006. But again, when we pull out the S-phase cells, we can use that population to define the level of IDU that's necessary for a cell to be in cycle, and then we can pull out a G0 population. And then phosphorylated histone H3, people use both the S10 and the S28. The S28 is slightly more accurate, but it defines this rare subpopulation here that's positive for phosphohistone H3 at a high level, and we already know from fluorescence studies that these are the M-phase cells, and so we're essentially just doing the same experiment here. So again, we can pull out G0, G1 here, S, G2, cycling cells versus G0 with the RB, and the M-phase cells. And we generally use the biaxial with iododeoxyuridine to define these. And so if you compare both the fluorescence and the mass cytometry experiments, you can actually essentially find all the same populations. So here it is by mass cytometry. Again, we can find IDU positive, high RB cycling cells, then RB low. These cells here that are completely negative, these are cells that are typically either dead or senescent. But we haven't completely defined this. But most of the G0 cells are actually sort of mid on a phospho-RB, but again, you're using the S-phase cells to define that population. And here's a really good example of the G0, G1 group, S, and G2, so you can get pretty distinct populations. And if you do the same experiment, so the same markers by fluorescence, they still will work by fluorescence. So if you detect IDU by fluorescence using an antibody to anti-IDU, just like you do BRDU, you can, again, define these same populations, like a cycling level of phospho-RB versus a non-cycling level, and then the same G1, S, G2 populations using the cyclin B. So it's not sort of a, this is a characteristic you could use for any kind of analysis. If you wanted to do a cell cycle by fluorescence and didn't want to use a DNA stain, you could do it this way as well. And you can see that this correlates really well with pyronin, but I'll show you more of that later. So here's just a group of the same cells. IDU was incorporated, and they were analyzed both by fluorescence and by mass cytometry. I think this was done in quadruplicate. And you can see that there's a little bit of a difference here in the G0 population, but otherwise the fraction of the cells lines up quite nicely. So this is a very good correlate to doing cell cycle by BRDU, Host, and pyronin Y, which was sort of the gold standard that I compared it to. And here's an example where we wanted to make sure that this was sort of biologically valid. So since we were working on cell lines, we thought, well, maybe it's just a size thing, or maybe there's some other reason why this is correlating. So we wanted to use this system where we'd know what phase of the cell cycle they were supposed to be in. So we started with normal resting T cells, stimulated them into cycle with PMA and inomycin. And so in this experiment, you expect them all to start off at 0 hours in G0, and indeed they do, regardless of which technique you use. There's a little bit of spray, but these may just be sort of activated T cells from the patient, but almost 100% of them are in G0. By 28 hours, you see the cells by fluorescence starting to enter S phase, start to see a little bit of a G2 population, and you see a lot of cells that have shifted from G0 into cycle. And again, by mass cytometry, we see essentially the same effect. By 28 hours, they're starting to enter S phase, and we see a big shift from G0 to cycling levels of phospho-RB. And by 48 hours, we see sort of a complete cell cycle distribution, both by fluorescence and by mass cytometry. And again, the fraction of these cells lines up pretty well by both techniques. And at this point, we've tried this in lots of different cell types. We've tried it in these peripheral blood T cells. We've tried it in whole mouse bone marrow. We've tried it in 5 or 6 cell lines. and in whole human bone marrow in the experiments I'm about to describe. So it's pretty robust, and there's a lot of data that I published in the cytometry paper if you want to take a closer look. So what can we do with this? So we wanted to focus on now using this as a systems level analysis. And what's cool is that really this only takes IDU, but that's not a channel that you were using anyway. And then at a minimum, just 2 more markers from there. So phosphorylated RB and cyclin B, and then if you want to do M phase, histone H3. So with only 3 of the channels that you would normally use for antibodies taken up, you can do a complete cell cycle analysis along with whatever else you wanted to do. So I'll talk to you about, you guys have already heard about SPADE, is that right? Okay, so I'll just go through it really briefly, and then how we use this to define the normal cycle, cell cycle distribution of human bone marrow, and then do briefly some comparisons of malignant and normal hematopoiesis. So again, the reason we use SPADE is that as you increase the number of parameters, the number of 2-dimensional plots expands exponentially, and you really can't look at the data here in 2 dimensions effectively. So we use SPADE. Essentially, if this is sort of a 3-parameter simplified experiment, you have areas of high density of cells, you have areas of low density. The first thing it does is even all that out by removing the high density areas. Then it clusters in n dimensions. Typically, we're using something in the teens in terms of dimensionality, just grouping like cells with other like cells. Then it takes these clusters of cells and links them together, again, like with like, so attaching them to their nearest neighbors in n dimensions to create a minimum spanning tree. Then once we have the tree, of course, the computer doesn't know what a T cell is or a B cell, so we ask it to color each of these groups of cells by their expression pattern. And we can pretty quickly see, well, these are the CD3 positive T cells. Here are the CD8 T cells, CD4 T cells. Similarly with B cells, we can use those markers to define them. And going through all this, you can get to essentially a complete description of all the immunophenotypes present in complex populations. And this, of course, comes from the science paper that we published. So here's an experiment I did in normal bone marrow. We were basically able to find all the cell populations we'd expect to see. We could define most of the B cell lineage, monocyte development, different subsets of T cells. Here you can just see, here's the CD3 positive T cells, CD15 positive granulocyte lineage, 14 positive monocyte lineage, B cells positive for 20. 235 defines this erythroid population here. 34 gives us our progenitors and stem cells. And once we've done that, now we can use those populations to look at the cell cycle state across these different groups. And so here we can just color the B cells here for their expression of the common defining markers. So we can find the pro-Bs have a little bit of 34, CD10 in the pro-Bs and pre-B1s, 19 in the pre-B2s, and 20 is only in basically the mature populations. Once we've defined these populations, I now have colored the tree. I've made the size distribution of the tree absolute. Now, when we typically display these plots, the size distribution has a minimum enforced so that the circles can't get so small that you aren't able to see the colors. Here, I've taken that away. So the size of the circle is sort of absolutely correlated with the number of cells in that population. And then in the inset, in the red, is the frequency of cells that were in each phase of the cell cycle. So here in G1, you can see that we see most of the G1 cells are present. It's hard to see here, but a lot of the really early pre-B2s have a high fraction of G1, and as well as in the pre-B2 population. And then when you get out into the mature cells, there's much smaller amounts of G1. Here in S, you can see that most of the S-phase cells are present in the pre-B2 population, which again correlates with sort of what we know about when B cells grow, and really no S-phase cells in the mature B cells, and sort of a similar pattern with G2. Interesting, there's very few G2 cells. This was something that was a little bit of a surprise when we started doing the cell cycle analysis. When you actually look at primary cells, the fraction of G2 is much lower than in cell lines, and probably this is because a lot of the G2 cells in cell lines are really sort of checkpoint arrested at a G2M checkpoint, as opposed to being in sort of a classical G2. And when you look at like embryonic stem cells, for instance, you also don't see a very large fraction of G2 cells. So this is what we believe is sort of a normal distribution. And we've done, this was just sort of a proof of principle experiment, but I've done the same experiment on a set of 5 samples, and we get essentially identical results, and they're really tightly correlated. So this effect appears to be real. We do the same thing in the erythroid population, again using CD34, CD71, CD235 to define the different groups of erythroid cells. Again, we color the tree, and we can see that most of the proliferation, both in terms of sort of G1 and S phase cells, is going on in the erythroblast population. We can see some more mature erythroid cells that are 235 positive and CD71 low, and they have a relatively low fraction of S and G1 cells. And again, there's some G2 cells, but they're pretty rare, consistent with the other findings. Again, looking in the myelocyte development, you can see that most of the proliferation in the myelocytes is coming in the promyelocyte and myelocyte stage. And again, mature granulocytes, mature myelocytes here have relatively low levels of S phase and G1 phase cells. So then we applied this to a subset of AML samples. We've expanded this analysis just very recently, but that data is sort of not fully analyzed yet. But this is sort of just one of the pilot experiments. Again, we did a very similar process. We analyzed all the cells together in the same experiment. This allowed us to define HSC early progenitor nodes, mature, and two different groups of very immature myeloid cells here. We could get T, B, and K, a mature myeloid branch, and sort of a promyelocyte branch here. This here is the tree for normal, colored for CD45. And then when we break down the different subsets of AML, here is just an example of two different patients. We can see there's some interesting differences. For one, AML sample 5 has a much higher relative expression level of CD34, whereas the CD34 positive cells in AML9 are not quite as bright. We can see that most of the AML5 samples are in this sort of CD34 positive branch, whereas most of the AML9 samples are in a slightly different branch. So we can see sort of distinct subtle differences in the immunophenotype. But what's more interesting is we can see this sort of branch of mature monocytes and mature granulocytes that's still quite expanded in AML sample 5. We don't really see that effect in AML sample 9. And then what's really interesting is that the distribution of S-phase cells across these different subsets is pretty different. And so here in AML sample 5, you can see that in the 34 positive branch, we have relatively high fraction of proliferation. As we get down to what I can tell you are the most immature 34 positive cells, that proliferation is a little bit lower. You can see a lot of proliferation is going on in a cell population that correlates with normal myelocytes. But then when you get out to mature granulocytes, you can see that the relative amount of proliferation is quite small. Here's this sort of expanded region of immunophenotypically mature monocytes, and you can see there's essentially no S-phase cells in that, even though this is clearly expanded, and these cells had 34 expression, so they are abnormal cells. So not all the cells in leukemia are proliferating the same, and we can see a very different pattern here in AML sample 9. There's this huge expansion of these immature proliferating populations in this branch with very low fraction of S-phase cells. And then most of the proliferation, even though it's still relatively low, is coming from this region here. We sort of scale the circle and circle plot. So actually here, a completely filled circle is only 20% S-phase. So these S-phase fractions are actually quite low. And then lastly, we can use this to look at how cells respond to chemotherapy, which is sort of the goal I had in developing the methodology. And what's interesting here is that when you subset cells by their cell cycle state and look at how they respond to a DNA damage treatment, in this case I'm using etoposide, you can see that most of the DNA damage response gets concentrated in the cycling cells, which we sort of knew. This is how these things work. But the difference is pretty striking here. So you can see that normally for H2AX, for instance, there is a little bit of a shift that normally happens in untreated cells as they go into S phase. This, of course, is the reason we have things like H2AX. It's not to deal with chemotherapy, but it's to deal with sort of damage to the genome that happens during normal replication. So you can see a little bit of that. But then when you treat them with etoposide, you can see this big shift. But you can see that the shift in terms of DNA damage when you look at the G1 cells here on this line is not all that dramatic, and the G0 cells have almost no shift in response to the etoposide treatment, whereas the shift in terms of the S-phase cells is quite dramatic, and you can see a big shift in DNA damage response here. See the same thing in ATM, some shifts here in CHEK2, phospho-p53. So this is also a very interesting way to sort of subdivide how cells respond to complex stimuli by cell cycle state. And again, it still leaves you plenty of other parameters left over to do other analyses simultaneously. So just in conclusion, this is sort of a validated methodology for using mass cytometry to assess cell cycle state. Again, this is published in Cytometry if you want to look it up. We were able to use this to do sort of systems biology of cell cycle in normal hematopoiesis. And essentially, we can use this to assess the proliferative state of hematologic malignancies, and that work is sort of almost completed now. And this allows us to combine cell cycle state with multiple other variables, and sort of my ultimate goal with this work is to be able to look at how cells respond to chemotherapy in vivo across all these different subpopulations and assess some of the hypotheses about how cells become resistant to therapies. And so the people that are most directly involved in this work, obviously Gary and Wendy as sort of mentors in the lab. Sean and Aaron kind of taught me the ropes on CyTOF. A lot of this was based off some work that Matt Clutter had done on cell cycle analysis. I get a lot of technical support from Rachel and Angelica. And then the SPADE people like Michael Lindemann and Sylvia Pulveritis and Peng Kui, who's now at MD Anderson, were obviously really instrumental in the kind of analysis. And then I work with a bunch of clinical collaborators to get these samples, Bruno Medeiros and Peter Greenberg in the hematology department, and Dr. Beverly Mitchell, who's the director of the cancer center here as well. So with that, I'll take any questions you guys have about the cell cycle methodology. Thanks. Mm-hmm.
Treat them, do you get different populations coming out?
Oh, yeah, that's interesting. That's not something we've done yet. I mean, mostly because my initial focus right now is just sort of looking at what does the cell cycle state start out as, and how does that correlate with different like subtypes of leukemia. But you really need a different clinical protocol, because you would have to really do those experiments, you have to get more bone marrow biopsies than you would normally clinically need. And so, when you want to get an IRB protocol to do extra bone marrow biopsies, it's a lot trickier. So we wanted to sort of get the baseline data first. So yeah, I don't have a great answer to that question. I can tell you when we've gotten sample sets where there's relapse samples and the correlated diagnosis sample, there's almost always a shift towards more immature immunophenotypes. And that's pretty widely observed. You can find other papers that describe that same phenomenon.
I have a potentially naive question. With fluorescence-based cytometry, what are the limitations with respect to the breadth of cell types you can look at? Is that one of the limitations, or can you only do one cell type and look at different phases?
I mean, there's a little bit, in some of the markers there are some subtle changes developmentally, so you sometimes have to do slightly different gates for different immunophenotypes, but there's really no limitation. In fluorescence, the biggest limitation is that a lot of these cell cycle markers, like the DNA intercalators, they're so bright, so they sort of take out like your whole laser. So that's one limitation. But if you can design the experiment properly, you can still do probably 5 or 6 other colors in addition to cell cycle analysis. And I've never found, I mean, I don't do a ton of fluorescent cell cycle analysis, but I've never found populations that you couldn't sort of analyze.
Okay, but you're not limited on the number of cell types you look at a given experiment, even with fluorescence?
No, I mean, certainly with mass cytometry, we can define, we just have the markers that allow us to define all sorts of different subtypes. And there's no subtype that we couldn't get cell cycle data from. And I don't see any reason why that would be a problem with fluorescence. The problem is just, like I said, these markers, like Host, for instance, is so bright. So like, when we use it on the 405 laser, like there's nothing, you can't put anything else on the 405 laser. And you still are even getting some interference into your 488 laser. So, those are the main issues with doing cell cycle analysis. I think if you wanted to do really high parameter cell cycle analysis, you could actually use the markers that I've defined here, because the cyclin B isn't as bright and the phospho-RB is not as bright. And so, you could use those parameters instead. and that might allow you to get more channels if you're doing like, you have like a 4-laser machine. Because, you know, even on a 4-laser machine, if you put packed blue or Host in there, it's really going to blow out some of your lasers. So you could try using this kind of approach and maybe be able to pack in more channels that way. So it's phosphorylated RB, but it's the serine 807, 811 phosphosite, which has previously been shown to correlate with the G0, G1 transition. I don't know if it's 100% perfect correlation, but it's very correlated with pyronin Y, which is sort of the gold standard. I didn't show you this, but if you do like in that experiment where I had both phospho-RB and pyronin Y, the R squared is like 90-some%. So it's very tightly correlated with pyronin Y. Whether pyronin Y is perfectly correlated with G0 to G1 transition, I think maybe one could have a debate about that, but I think it's as good as what you could do fluorescently.
Sorry to cover this, but can you recognize aneuploid cells, and has that been found any of your analysis?
I've not seen anything that I thought was clearly an aneuploid cell, but you know, in the patient subtypes that I'm doing, they all have clinical diagnostics, so I know what their karyotype is. So I mean, obviously some of them have like a trisomy or something, but you can't detect a trisomy by any sort of fluorescent technique. So I have never really seen what it would look like on, you know, a sample set that had 70 chromosomes or something like that. Kara might be able to. I guess Kara's not really done cell cycle. You've used samples that had 70 chromosomes, but.
Yes. But I can't say that the DNA, like looking at the DNA staining, can discern hyperdiploid.
There are some published reports, yeah, by fluorescence where you can see like really hyperdiploid cells look different, but the pattern's still the same. It's just like the whole curve shifts a little. So, yeah, I don't anticipate